Hi, my name is Phillipa Beale. I'm a veterinarian and an ecologist, and I'm going to be talking about the effects of temperature on feeding by marsupial folivores. So finding and obtaining food is something that's pretty fundamental to an animal's survival. It's what allows them to perform all the basic functions from reproduction to growth, moving around. So if they're unable to find and access enough nutrients, then they're unable to perform all of these functions. So for marsupial folivores, we're talking about eucalyptus leaves. So for them to be able to find and obtain enough food, we need to be able to really manage our eucalyptus forests well into the future. So the traditional view is that the amount of food that an animal can find is what limits the amount of output activities like reproduction or movement that animal can do. So energy in from feeding leading to these energy output activities. And then ultimately all of that energy gets lost as heat. And there are plenty of situations where this is the case. But there are also plenty of situations where this assumption is fundamentally flawed, where food is abundant in the environment, and yet animals don't simply eat more and then do more output activities. So what's going on in these cases? Well, Speakman and Kroll argued that animals are actually shuttling between two states. The first, where energy input is limited by the environment, and the second, where energy input is limited physiologically. So they came to this conclusion through a series of experiments with laboratory rodents, in which they thought, what is the most energetically expensive thing that a mammal can do? And it's probably lactation. So they got some mice, they made it so that they were lactating, and they measured how much the mice were eating. So when they were lactating, they ate a bit more food. And then they tried to artificially increase their energy expenditure even more. So they artificially increased their litter size. They made it so that they were pregnant while they were lactating, and they made it so that they were exercising while they were lactating. And in all of those cases, the mice didn't increase their food intake any further, even though their energy expenditure was greater. The only things that they could do to make them increase their food intake were put them in the cold or shave them. And in both of those situations, the mice increased how much food that they ate, they produced more milk, and they reared larger offspring. So what these two scenarios have in common is that they allow the extra body heat that's produced through the metabolic processes involved in lactation to be more easily dissipated into the environment. So they termed this heat dissipation limitation. It's the idea that the rate at which an exothermic metabolic process can take place is limited by the rate at which the heat that's produced from that process can be lost into the environment. So of course, this is pretty relevant when we're thinking forward with our changing climate. We're expected to see more frequent and more severe extreme weather events like heat waves. So this is a map of Australia from the Bureau of Meteorology in our particularly hot summer of 2019-2020. You can see that most of Australia in this map is pushing well into the high 30s, if not into the 40s. So what we're talking about though is, what does this mean for marsupial folivores? Most of the studies looking at how climate change will affect this plant herbivore interaction have looked at it from the perspective of the plant. So higher carbon dioxide levels in the atmosphere are likely to cause high levels of fiber in the leaves, higher levels of the plant secondary metabolites or plant toxins, and also lower levels of protein. But what does it do to the animal? So I studied common ringtail possums and common brushtail possums to answer these questions. So common ringtail possums are cuter. They are smaller, typically between half a kilo to just over a kilo in size. They're more specialized, so they prefer the monocalyptus group of eucalypts, and they are more difficult to catch. While brush-tail possums rely more heavily on their personality, they are a bit larger, so more like 1.5 to up to 4.5 kilos, and they are less specialized. So sometimes you might find them eating from your veggie patch, but they prefer the Symphioma group of eucalyptus leaves, and they're highly food motivated, which made them a lot easier for me to catch. So we're going to ask the question, how will warmer temperatures impact the plant herbivore interaction from the herbivore's perspective? So first things first, we need to know what is a warmer ambient temperature for a marsupial folivore like a possum. So the way you assess this is using respirometry. What that looks like is putting an animal inside of a temperature controlled chamber and measuring how much gas exchange is occurring. So how much oxygen is getting used up and how much carbon dioxide is getting produced. And from this, when the animal is at rest, we can infer their basal metabolic rate. So how much energy is an animal consuming just to maintain its core body temperature? And you do this across a range of different ambient temperatures. So there is a zone of ambient temperatures where the amount of heat from the body that's being lost into the environment can be easily counted through changes in posture or insulation, like piloerection, or how much blood flows in the skin. And that's called the thermoneutral zone. So in this zone, the metabolic rate doesn't get adjusted to counter gains or losses in heat from the environment. It's low and constant. This is bounded by an upper critical temperature and a lower critical temperature. And in possums, in ringtail possums that I tested this in, I found that the lower critical temperature of the thermoneutral zone is around 18 degrees, and the upper critical temperature is around 26 degrees, which might seem relatively low, but remember that they are nocturnal, so most of their activity is in the nighttime, the cooler part of the day. So as you move towards cooler temperatures below the lower critical temperature of the thermoneutral zone, metabolic rate has to be increased to counter the increased rate of loss of body heat into the environment. So that means the amount of energy an animal is expending just to maintain its core body temperature is also increased as metabolic rate rises. Somewhat paradoxically, above the thermoneutral zone, you also see an increase in the basal metabolic rate of the animal. This is thought to be due to the energy involved in like respiratory muscles used for panting, things involved in evaporative cooling mechanisms. But it's a much shorter, sharper increase, and typically endotherms don't cope as well with the hotter end of the temperature spectrum. So we're talking about food intake, and as you can imagine, where metabolic rate and hence energy requirements are increased, food intake is also higher. So from at the cooler end of the temperature spectrum, we see higher food intake, and then moving towards the warmer end of the temperature spectrum to counter, moving towards the warmer end of the temperature spectrum, the food intake decreases. However, even though the metabolic rate is low and constant in the thermoneutral zone, and even though it's increased above the thermoneutral zone, we still see further decreases in food intake at these warmer temperatures. This is something that's observed really commonly in domestic animals, and it affects their outputs. So grower pigs will grow less big in warmer ambient temperatures. They'll eat less. Your dairy cattle will produce less milk. Broiler chickens won't grow as big or as fast in warmer ambient temperatures because they won't be eating as much. So what's happening here? Well, in between this feeding and energy output step, there's another step. So the food that the animal eats has to be digested and absorbed. And some part of that energy stored in the food during these digestive and absorptive processes gets lost directly as body heat. So remember, in the case of marsupial folivores, we're thinking about eucalyptus leaves, but they contain the same macronutrients that our food contains. So they contain carbohydrates, fats, and proteins. And marsupial folivores are remarkably good at looking at the range of leaves in their environment and making nutritional decisions based on the composition of those leaves. But for us, it's a little bit hard to visualize. So I'm going to represent it like this, something very easy for us to visualize. So carbohydrates, fats, and proteins all contained within a burger. And looking around the environment, you would have a range of foods with different compositions, relative compositions of these nutrients. So some have more protein, some have more carbohydrates. So while digesting and absorbing these macronutrients, about 3% of the energy that's contained in fats, about 10% of the energy that's contained in carbohydrates, and about 30% of the energy that's contained in proteins gets lost as heat. This goes by many names. It's sometimes called the heat increment of feeding or diet-induced thermogenesis. So what that means is if you had two meals, both of the same composition, but one was larger than the other, then the larger meal would result in more diet-induced thermogenesis than the smaller meal. If you had two meals of about the same size, one had more protein, one had more carbohydrates, then the higher protein meal would result in more diet-induced thermogenesis. And if you had two foods of different compositions and different sizes, you could even end up with a smaller meal with higher protein, resulting in as much diet-induced thermogenesis as a larger meal that contained more carbohydrates. So since digesting and absorbing macronutrients is an exothermic metabolic process, could it be that heat dissipation limitation also plays a role in constraining what feeding decisions the animals are able to make? So to test this, I acclimated 12 possums to either 10, 18 or 26 degrees, remembering that 10 is below the thermoneutral zone, 18 being around the lower critical temperature, and 26 around the upper critical temperature of the thermoneutral zone for possums. So I acclimated them to those temperatures for a week, and then they were offered two diets at the same time, one that was high in protein and one that was high in carbohydrates and low in protein. So they were able to choose how much of each diet they ate. And then all of the animals were exposed to all of the different temperature treatments. So one way of visualizing this type of study is using the geometric framework of nutrition. So in this, we plot different nutrients against each other. So on the y-axis here, we have non-protein dry matter intake. So that was the carbohydrates and the fats. And on the x-axis, we've got protein dry matter intake. And then the relative proportions of these nutrients is in each diet is represented by a nutritional rail. So the low protein diet you can see is a lot closer to the non-protein y-axis, and the high protein diet you can see is a lot closer to the protein on the x-axis. And so if you were eating just the low protein diet, you could eat anywhere along that nutritional rail, but because you're only eating that food, you can't change the relative proportions of these nutrients. So you're just going, if you ate less, you would be closer to the origin. If you ate more, you would be further out along that rail. So in eating those two foods, you could eat a diet composition that is anywhere between those two nutritional rails. If the possums were choosing purely at random, on average, you would end up with something along this equal intake scenario. So that would indicate that they're not really choosing, they're just at random eating a bit of one, a bit of another. But what I found was that at 10 degrees and at 18 degrees, the possums ate along approximately the same nutritional rail. So they mixed their diet to make something of the same composition. At 18 degrees, they ate slightly less than at 10 degrees. But when they ate at 26 degrees, they mixed their diet to make something that was lower in protein. This might seem like a relatively small change in diet, but it's really important not to underestimate how small changes in diet can affect populations. These are called multiplier effects. So it's really important not to underestimate the importance of these really small or seemingly small changes in nutrition. So just a relatively small change in digestible nitrogen, which is an indicator of the amount of protein in the diet, possum breeding success started to decline dramatically below about 0.4% digestible nitrogen. And these things can have really large effects at the population level. So remember, we're not actually talking about burgers. What we're talking about is eucalyptus leaves. So I wanted to see how what we offered in the lab related to what was available to possums in the field. So we collected leaves from all of the Eucalyptus melliodora trees that were on Black Mountain in Canberra. Canberra is where these studies took place, where the possums were from. And the leaves were analysed in terms of their available protein and carbohydrates. So choosing from all of the trees on all of the Eucalyptus melliodora trees, they could eat anything within these polygons. So we've got available protein on the X and carbohydrate percentage on the Y. So they couldn't choose to eat anything that falls outside of these polygons. And we also broke this down into old leaves in the orange sort of color, young leaves in the blue, and leaf tips in the yellowy greeny color. So what we offered them actually fell outside of this. So the low protein food was lower in protein than any of the leaves that were available to them. And the high protein diet was lower in carbohydrates than any of the food that would be available to them if they were eating Eucalyptus melliodora from Black Mountain. But the diets that the possums mixed for themselves in choosing from these two options fell within what they would have been able to consume in the wild, eating old leaves and young leaves. So they wouldn't have been able to mix a diet that looked like leaf tips, but leaf tips are not available for all of the year. So this seems pretty reasonable. And I didn't measure what it would look like if they were eating hot chips out of the bin, but I assume it would be pretty high in carbohydrates. So something eucalyptus leaves have that burgers do not, or at least should not, is plant secondary metabolites or plant toxins. And these act as feeding deterrents or anti-nutritive compounds, and they're really important in determining feeding decisions in marsupial folivores eating eucalyptus leaves. So I wanted to know what would be the combined effect of temperature and plant secondary metabolites on macronutrient selection by marsupial folivores. So in this study, I just used two temperatures, 10 degrees and 26 degrees, to see whether they changed their macronutrient balancing at 10 or 26 degrees. When there was no cineol was the plant secondary metabolite, the plant toxin that was used in this study, or when there was. So when cineol wasn't added, we saw that at 26 degrees, the possums chose to mix a diet that was lower in protein than at 10 degrees. And when it was 26 degrees, they also ate less than at 10 degrees. So that's pretty similar to in the first study. And then when I added cineol, a plant toxin, a known feeding deterrent for brush-tailed possums, to both diets, they ate significantly less, which was not a surprise. But they also ate along about the same nutritional rail, and they ate less at 26 degrees than at 10 degrees. So what that tells me is there's some interaction or some interaction between how temperature affects nutrient balancing when there is a plant toxin and when there's not. Also, when cineol was added, they ate a diet that was higher in protein compared to when relatively compared to when the diet did not contain cineol. So Jesse Au and colleagues showed that when possums were eating diets that were higher in protein, they were also able to eat more cineol. So the light colored bars are the higher protein diets, and you'll see that across all these different cineol concentrations, they consumed more food when the diet contained more protein. So protein is really important for metabolizing plant toxins, plant secondary metabolites. So we can see that there are effects of warmer temperatures on feeding, that there are changes in macronutrient selection with changes in temperature towards lower protein diets in warmer ambient temperatures, and that there is an interplay between temperature, nutrients, and plant toxins in determining how macronutrients are balanced when folivores are making their feeding decisions. So in talking about plant toxins, there's an additional part to this process. So the detoxification processes when breaking down those plant toxins that animals ingest are also exothermic metabolic processes. So they also result in some heat production. So it could be that heat dissipation limitation plays a role in plant secondary metabolite or plant toxin detoxification. So this is called temperature-dependent toxicity, which is a term that was coined way back in 1959 by Keplinger. So in these studies, they used laboratory rodents and drugs, and they exposed the laboratory rodents to various housing temperatures, and then they fed them a whole suite of different drugs. And what they were looking at was the LD50. So what dose of the drug was required to kill half of the population. So a lower LD50 means a lower dose is required to kill half of the population, so it's more toxic. And a higher dose means that it's less toxic. And what they found was that at warmer housing temperatures, a lower LD50 was found for a range of drugs. So at warmer temperatures, they were effectively more toxic. But we're really interested in wild herbivores eating toxic plants. And this has been looked at in some other wild herbivores. So the white-throated wood rats are found in the Mojave Desert, and they eat toxic creosote bush. And Kernath and colleagues have looked at whether temperature-dependent toxicity applies to these wood rats. So they conducted a persistence trial in which they housed the wood rats at different temperatures. So the cool temperature being 21, the warmest temperature being 29 degrees Celsius. And then they offered them a diet that had creosote resin in it. What they found was that the cool temperatures, the animals persisted in the trial, but so animals were removed when they lost 10% of their body mass. So at the warmer temperatures, they were removed more rapidly. So they were able to persist on this diet if they were housed in cool temperatures, but they weren't able to maintain condition and persist in the trial if they were housed at warm temperatures. And that's because the prevailing rhetoric, at least, is that animals that eat toxic plants would rather starve themselves than intoxicate themselves. So this scenario doesn't actually happen. They're really good at regulating their intake around certain set points. So in this study by Stapley et al., you can see that as a gentsenone, another plant toxin, concentration increases in the food, the intake of the food decreases so that they regulate around a certain maximum threshold. So I ask the question, do warm ambient temperatures limit plant toxin tolerance in marsupial folivores? I think this is a little bit counterintuitive. So I'm just going to go through it a bit slowly. So from what we discussed earlier, we expect to see a decrease in food intake at warmer ambient temperatures compared to cooler ambient temperatures when there are no toxins involved. So that's their preferred intake at those temperatures. If there were no effect of ambient temperature on intake of a diet that contained toxins, then we would see that it's always less than their preferred intake. And since they're regulating around a certain tolerance threshold, if there were no effect of temperature, it would just be the same across all the temperatures. And if there was some effect of temperature, then we could see anything different from that. So it could be that they increase their tolerance with warm temperatures, decrease their tolerance with warm temperatures. We see some change in their intake of the food that contains the toxins with changes in ambient temperature. So anything different from this, indicating a change in tolerance to the toxins with a change in ambient temperature. So to test this, I captured 12 ringtail possums and I acclimated them to 10, 18 and 26 degrees once again for a week. And then at the end of the week, I offered them the choice, not a choice, sorry, I offered them leaves that contained either high sideroxylenol levels, so another plant toxin that deters feeding in ringtail possums. So I fed them Eucalyptus melliodora with high sideroxylenol levels, or I fed them Eucalyptus melliodora with low sideroxylenol levels. And then in randomized crossover design, all of the animals were exposed to all of the different diet and temperature treatments. I did a similar study with brush-tailed possums as well, but using artificial diets and the plant toxin flavanone. So for the ringtail possums, I'd have to weigh in the bunches of leaves before feeding and then weigh them out to measure how much the animals had actually eaten, which sometimes they made a little difficult. For example, they might stuff them into their nest box like this, make a cozy little bed, or they might decide they don't want to eat those leaves and just strip them off, and then they'd look at you like this and you'd be having to pick them all up. So what I found was that when the ringtail possums were fed the high sideroxylenol leaves, they reduced their intake compared to when they were fed the low sideroxylenol leaves, and they reduced their intake of the high sideroxylenol leaves further at warmer ambient temperatures. And I found something similar in brush-tailed possums. So they reduced their intake of a high flavanone diet when ambient temperature was warmer. If I only exposed the possums to the different temperature treatments for one day, then this effect was greatly reduced. So you can see in the ringtail possums, the intake of the high sideroxylenol leaves, the high toxin diet was the same across the different temperatures, while the intake of the low toxin diet decreased as temperatures became warmer, so at 26 degrees compared to at 10 degrees. And the effect was also less when exposed for 24 hours in the case of the brush-tail possums. So do warm ambient temperatures limit plant toxin tolerance? The answer is yes, but in an exposure time dependent way. So why might this be the case? Well, if we think about what's actually happening inside of the animal, if they have a meal that contains the toxin and then nothing else takes place, it's absorbed, enters the bloodstream, and then the blood concentration just stays at that level forever. But of course, that's not what's going on. So these plant toxins are metabolized in the liver, usually by the family of enzymes called cytochrome P450 enzymes. And the liver is one of the most thermogenic organs in the body. So once the toxin has been absorbed into the blood, some detoxification processes happen, and then the blood concentration is reduced. So across a series of meals, you'll have this certain regulation around certain levels of intake and detoxification so that the blood concentration remains relatively stable and is regulated below maximum level thresholds. So we're familiar with this from, say, the 1 drink an hour rule so that you can drive. How if you took your toxin too frequently and the detoxification processes couldn't take place, and your blood concentration would increase. Likewise, if your detoxification processes weren't happening well enough, then your blood concentration would increase above that maximum level you're trying to regulate below. So we're going to ask the question, does temperature change the hepatic detoxification capacity? And to test this, I used a proxy for plant secondary metabolites for the plant toxins called alfaxan. So alfaxan is a veterinary anesthetic. So it's also metabolized almost exclusively by cytochrome P450 enzymes in the liver. So this means that I could use it as a good indicator of how the plant toxins would be metabolized. So I once again exposed possums to 10, 18, and 26 degrees. And then I gave them all the same dose of alfaxan and as the proxy for that plant secondary metabolite intake. And I did what's called a sleep time assay. So I anesthetized them using alfaxan, and then I measured how long it took before they woke up. So a shorter amount of time means that they're detoxifying the alfaxan a lot faster, and a longer sleep time means that they are detoxifying the alfaxan a lot slower. So here's what that looks like. This is not the best example. So this possum, you put them on their back, give them while they're asleep, and they don't much like being on their back. So when they have detoxified or cleared enough of the alfaxan from their system, they flip themselves onto their side. And then that's the end of the assay. So what I found was for both brushtail and ringtail possums, when acclimated to those temperatures to 10, 18, and 26 degrees for a week, the functional clearance time, the time for which they slept, was a lot longer at 26 degrees compared to at 10 degrees. So at warm ambient temperatures, that means they cleared the alfaxan from their system through detoxification processes in the liver a lot slower when it was warm compared to when it was cold. But if I only exposed them for 24 hours, that same effect wasn't seen. So there was no difference in the functional clearance time at 26 degrees compared to 10 degrees if they were only exposed for a day. So does ambient temperature change detoxification capacity? Yes, in an exposure time dependent way. So what we have so far is at warmer ambient temperatures, if exposed for long enough, possums reduce their hepatic detoxification rate, and in turn they reduce their plant toxin intake. And they also change their macronutrient selection, and there's some influence or some interplay between macronutrient selection and toxin intake. There are also some other effects of other interactions between diet and temperature. For example, plant toxins can influence in thermosensing, and it can also influence energy metabolism in the mitochondria. But I won't be talking about them because we're interested in what's happening in the field. So we're going to talk about koalas now. So the population of semi-wild koalas in Phillipa Island, and they were collared with radio collars, radio tracking collars, and microphones, so that they could be tracked to whatever tree that they were in, and the microphones were used to monitor if they were feeding or not. And this was related to a whole bunch of feeding studies in the lab, so that we could determine exactly how much they ate of what tree and when. And there were also temperature data loggers recording what temperature it was while they were feeding. And the trees that they were feeding from were all, the leaves from these trees were sampled, and they're all measured, all tested for their FPC concentration, which is another group of plant toxins that deter feeding in koalas. So on warmer days, the daily intake of food was lower. And that's not a huge surprise, as we talked about before. But when we dig a little deeper into what's causing this reduced intake of food, what we see is that rather than decreasing the number of meals, the koalas are actually decreasing how much they eat per meal. So in warmer ambient temperatures, the size of the meal is a lot smaller than in cooler ambient temperatures. And if this is broken down even further by FPC level, so this maroon bar is the high FPC leaves, the yellow is the low, and the red is the mid-level FPC leaves. What we see is at cooler temperatures, they actually ate more of the high FPC leaves, but beyond about 18 degrees, they never chose to eat from trees that contained high levels of FPCs, instead always choosing low or mid-range FPC level trees. So you might wonder, why would they ever choose to eat the high toxin leaves? Well, the amount or the concentration of FPCs in the leaves and the concentration of available nitrogen, remembering that's a good indicator of the amount of protein in the leaves, are highly correlated. So higher FPC levels, those leaves usually have higher available nitrogen levels or higher protein levels as well. So that means at these cooler temperatures, they were able to access more protein than at the warmer temperatures by being able to tolerate higher FPC leaves. Looking forward, what does this mean? So if an animal is, if a marsupial folivore is in a forest choosing which trees to feed from, there's a range of different toxin levels and nutrient levels in all of the different trees. And so at the moment, there might be lots of different trees to choose from, and they might just avoid the ones that have really high toxin levels and choose to feed from the ones that have lower toxin levels. But when temperatures are warmer, the effective toxicity of trees that have higher toxin levels is increased because of changes in the physiology at the animal level. So it means they have less options to choose from. So as we get even further into higher ambient temperatures, those choices are even further reduced. So what that means when looking into the future into how we can best manage our forests into the future, what we need to do is ensure that they have the diversity of trees to be able to make the choices they need to make under different temperature regimes. So then they can make those feeding decisions to be able to meet their nutritional requirements, even when they're physiologically constrained by the heat. So thank you for listening.